Hello, my name is Joe Rucker, and I'm the Vice President of Research and Development at Integral Molecular. I want to thank the organizers, particularly Ken Simmons, for helping assemble this really interesting meeting during these very challenging times. This session is going to focus on transporter proteins, which I don't think have gotten the attention that GPCRs and ion channels have, but these are very compelling targets with some unique challenges, which I hope to actually get you excited about. For those of you unfamiliar with Integral Molecular, we are the leaders in antibody discovery against complex membrane proteins. Located in Philadelphia, we have close to 20 years of experience in the discovery and characterization of antibodies against the most challenging membrane protein targets. We've worked on thousands of different membrane protein projects and have experience with most classes of membrane proteins, including GPCRs, transporters, and ion channels, which are the topic of a lot of the sessions of this meeting. As an antibody discovery company, Integral Molecular has a greater than 95% success rate in discovering antibodies against membrane protein targets. Underpinning this success is a suite of technology platforms tailored for membrane proteins and actually offered as independent products and services. While I will discuss these within the talk, I will briefly introduce them here. Our lipoparticle technology. Lipoparticles are virus-like particles produced in living cells and presenting high levels of specific membrane proteins in their native structure and orientation. Lipoparticles are used throughout the entire antibody discovery process, really from the immunization and discovery side, all the way through characterization and engineering. Shotgun mutagenesis is a platform for mapping how antibodies interact with their targets, the antibody's epitope. Briefly, we create comprehensive mutational libraries across an entire protein, usually alanine scans, express these variants in living cells, and then interrogate those cells, those variants, for antibody binding by flow cytometry. And the loss of binding can be used to identify those residues that are critical for the antibody to bind, in other words, the epitope. Finally, our membrane proteome array is 6,000 human membrane proteins arrayed and expressed in living unfixed cells. This MPA can allow us to look for antibody binding across the entire human membrane proteome and look for off-target binding, which is a major liability during therapeutic development, as well as for target discovery. Now, while we tend to think of complex membrane proteins, these multi-spanning membrane proteins like GPCRs, ion channels, transporters, as being really good drug targets, what we really mean is that they're very good drug targets for small molecules. If you look at antibodies, there are only 3 FDA-approved antibodies that are against complex membrane protein targets. So why is it actually so hard to generate antibodies against these types of proteins? Well, there's a number of reasons. One is, of course, poor immunogenicity, in part because many of these proteins have limited extracellular domains, and also sometimes they can be quite highly conserved within rodents, within mammalian species, making it hard to get a good immune response. And of course, membrane proteins can be just really challenging to work with. Oftentimes, they don't actually express very well, or if you do express them really well, they kill the cells, so they're very toxic. And also, really, the key thing is that in order to work with membrane proteins in their native structure, you need to work with them within a lipid membrane. If you take away that membrane, the proteins fall apart, and you're not going to get good antibodies. So let's go on to more specifics here. The transporters. Transporters are the proteins that are responsible for mediating the transfer of diverse nutrients and metabolites across the plasma membrane of cells. And the ligands for these transporters are really diverse. It's a lot of stuff. It's amino acids, it's metal ions, lipids, neurotransmitters, and sugars, as we'll see in this talk. There's really two major families of transporters. There's the SLC family, and there's the ABC transporter family, which is actually rather small. The SLC transporter family is actually one of the largest families of membrane proteins. It's over 400 members. And unlike GPCRs, where all the members of the family share a common topology, they often share common signal transduction mechanisms as well. The SLC family, superfamily more precisely, is really defined by their function as transporters. And the different family members within this superfamily can differ in topology and can actually all differ by mechanisms as well, and of course differ by the types of molecules that are transported. Now, different tissues in the body have really different requirements for metabolites and nutrients. And this is reflected in the repertoire of transporters they express. And in many ways, the different transporters that are expressed on a given tissue really can be very specific for that tissue. And this figure gives examples of different tissues and also the repertoires of transporters that are expressed in those tissues and the diseases that are associated with these particular transporters malfunctioning. And you can see there's a lot in the brain, there's a lot in different metabolic organs, in muscle, and really all organs not represented here, but a lot of immune tissue have very specific transporter requirements. And really all of this underscores the fact that that transporters really are very, very intriguing therapeutic targets. And from an antibody perspective, we really think they're an underexploited class. So let's get to something more specific here. So SLC2A4, which I'll refer to a lot as GLUT4, is a member of this SLT2 family of glucose transporters. There's a total of 14 members. And the role of these transporters is to transport glucose from the outside of the cell to the inside of the cell, where the glucose or other sugars can be used as fuel to produce ATP. This is a structural model of GLUT4 based on actual structures of other SL2 family members. And at Integral, we tend to refer to this as an iceberg. Most of this protein is embedded in the membrane, and really only a small portion of it is expressed in the exofacial part of the membrane, where it can be accessed from by antibodies. The largest loop is 29 amino acids, and some of the other loops are actually quite a bit smaller. So what does GLUT4 do? GLUT4 is expressed in adipocytes, fat cells, and also in smooth muscle. At resting blood glucose levels, which is actually relatively low, GLUT4 is expressed in these cells, but primarily located in these intracellular vesicles that are within the cell. After a meal, as your blood glucose levels begin to rise, insulin is released from the pancreas. And then insulin then activates insulin receptors on these different cells, the adipocytes and the muscle cells. And this activation of the insulin receptor causes a rapid translocation of GLUT4 from these intracellular vesicles to the cell surface. At the cell surface, this GLUT4 allows for the rapid removal of glucose from the bloodstream, which then, of course, gets converted either into fat in adipocytes or glycogen in the smooth muscle cell. And then this, of course, allows the blood glucose levels to return to a resting state. Insulin resistance is effectively the inability of insulin to stimulate GLUT4 translocation to the plasma membrane, and thus not allowing glucose to be removed from the bloodstream and causing, of course, hyperglycemia. This is effectively type 2 diabetes in a nutshell. When we started this project, there were no antibodies against the extracellular domain of GLUT4, and transporter translocation had to be monitored by indirect means. And we wanted to change that. So how does integral molecular make antibodies against these complex membrane proteins? We start with the protein of interest. We optimize the antigen, incorporate it into our lipoparticle technology. The immunizations we normally do are lipoparticles plus genetic immunization such as DNA, although I will say for GLUT4 we only did a lipoparticle immunization. And then we like to work in divergent species, and our species of choice is generally the chicken. Why chickens? Chickens are phylogenetically distinct from mammals and allow you to access antibodies against highly conserved antigens such as GLUT4. Once we have a robust immune response, we convert to an immune phage library and use phage display coupled with deep screening in order to find those rare antibodies that we're looking for. These could be agonists or antagonists, antibodies with very particular specificities. And then once we have the antibodies, we have lots of downstream capabilities as well for engineering, for humanization, and affinity maturation. And really the end goal is to have a best-in-class panel of antibodies that we can be used for whatever application you're looking for, particularly therapeutic applications. And as mentioned, we have a greater than 95% success rate in discovering antibodies against these complex targets. So how did we isolate antibodies against GLUT4? We immunized with a chicken with lipoparticles. We were able to get a nice serum response. We converted this to an immune phage library, did phage display, pulled out actually a fairly modest number of antibodies. This was actually a very small level of screening. We just screened a small number, pulled out 80 antibodies, and then did some deep characterization of 4 antibodies. I should note that these antibodies are interesting. Chickens in general have somewhat longer CDRs than mouse or human antibodies, and the antibodies that we chose to characterize definitely fell in that category of somewhat on the longer HCDR3 length. So the first thing we did is look for specificity, and these antibodies are specific for GLUT4. What we did is we looked at essentially total GLUT4 as well as cell surface. Of the 4 antibodies we did deep characterization on, 3 of them were seeing the GLUT4 at the cell surface. 1 was actually against an intracellular epitope. These antibodies in general saw mouse GLUT4, but did not see the closely related human or mouse GLUT1 paralog for GLUT4. The one exception to this is that intracellular antibody LMO43, which was cross-reacted between GLUT4 and GLUT1. And we'll talk a bit more about LMO43 because it's an interesting antibody. These antibodies were all tight-binding antibodies. We're able to use our lipoparticles in a biosensor assay to determine the association and dissociation rates. And all of these antibodies bound to nanomolar or subnanomolar. And one of the antibodies, LMO48, was really at the limits of detection and is very much a picomolar binder. We also wanted to dig a little bit more deeply into the membrane proteome array. The membrane proteome array, as mentioned, is 6,000 human membrane proteins expressed on living unfixed cells. We can probe the binding of the antibody to all of these overexpressed membrane proteins using flow cytometry and determine the off-target specificity binding of the antibody of interest. Our library contains 94% coverage of the membrane proteome with a very high coverage amongst transporters, ion channels, and transporters and GPCRs. The GLUT4 antibodies were highly specific, with the exception of LMO43, which had GLUT1. We did not see any significant off-target binding. Most of this is just noise. I will say for LMO52, one of these sort of targets binding in noise wasn't a binder. It was actually an off-target binding to an intracellular protein, Notch1, which turned out to have a shared epitope. with GLUT4. So a shared extracellular epitope with an intracellular epitope. I should note that much of this work was published in a PNAS paper in 2018, although I'll show some new data at the end of this presentation as well. The most interesting thing about these antibodies that we pulled out is that they're state-specific. Transporters in general have to exist in multiple states in order to allow their transport mechanism. They will have an outward open state, which can bind glucose on the outward part of the membrane, and then they can flip into an inward open state where it releases glucose into the intracellular milieu, into the cytoplasm. There are numerous ways of locking receptors into specific states. You can use pharmacological reagents like cytochalasin B or fluoretin. You can use mutations, which will lock it into an inward open or an outward open state. And what we discovered is that some of our antibodies were actually state-specific. You can lock receptors into an inward open state using cytochalasin B or this mutation here. And what we saw is that LMO43 was still able to bind to this lock state, while LMO48 was not able to bind into the lock state, and LMO59 was able to bind under both conditions. On the flip side, LMO43 was not able to bind in the outward open lock state. LMO48 was able to bind on that LMO lock state, but once again, LMO59 was state independent. So LMO43 binds to this inward state, and LMO48 binds to this outward state, which is really quite interesting quite interesting properties. So how does all of this work? We used our shotgun mutagenesis epitope mapping technology to figure out exactly what the mechanism of action of these antibodies were. Briefly, to reiterate, we do a comprehensive alanine scan across the entire protein, express in living cells, measure the binding of the antibody, measure the binding of the antibody by flow cytometry. We can find those mutations which knock out binding of the specific antibody, but not of cell surface expression of the protein in general. And we can map these onto a structure and understand exactly how that antibody is interacting with the target of interest. And we do a lot of this epitope mapping at Integral Molecular, and it's very, very successful, and often matches with things like crystallography when you can get that type of gold standard structural study. So in red are the epitope residues for these different proteins. You can see LMO43 binds to this intracellular intracellular helix here. LMO48 binds to an extracellular region here. It actually binds two residues that are on somewhat different loops. And then LMO52 binds onto the large loop of GLUT4. So why are these epitopes state-specific? And let's focus on LMO48. We can map these epitope residues. Once again, these are the epitope residues in red here on an outward open model and an inward open model. And once again, these are based on actual structures of closely related glucose transporters. And you can see that these epitope residues really change their orientation and their relative distance to each other in these two different states. There's also some other changes that go on in this structure, and so we can't rule out some steric occlusion as contributing to this state-specific effect. But really, this epitope explains the state-specific nature of LMO48, and you can see some more details once again in the published work. The fact that these antibodies are LMO48 is state-specific also suggests that this should be acting as an inhibitor of GLUT4-mediated glucose uptake, because in order for this glucose uptake to happen, it needs to go through these different alternating states, and LMO48 binding of it will lock it into that outward open state. So what we have here is that we can do a glucose uptake assay. In the absence of antibodies, you get good glucose uptake. In the presence of LMO48, you can get robust inhibition, which is dose dependent, while the other antibodies, control antibodies, show no inhibition whatsoever. We also see inhibition with cytochalasin B, and also when we run the assay in the presence of GLUT4, which is in a locked conformation. We were also interested in being able to use these reagents because they're very high, these antibodies, because they're very high affinity, to actually look at glucose transport in living adipocytes. So these are experiments that were done on murine 3T3L1 adipocytes using L148. This is work that was done in collaboration with the University of Cambridge, Daniel Fazakerli and Dougal Norris, who have really been great collaborators. What they showed is that in the absence of insulin, they see no GLUT4 that's on the cell surface of these adipocytes. But as they at increasing levels of insulin, they get increasing translocation, which can be easily detected by our antibodies. Their goal in all of this is to actually use these type of assays in working with RNA inhibition to look for novel regulators of GLUT4 translocation. And what you have here is you have essentially this dose response of insulin from no insulin to high insulin, and then knocking out these well-known regulators of translocation, and then you can see reduced regulation. And these are known regulators, and what they're doing is looking for novel regulators of translocation, which are potential druggable targets for diabetes and for insulin resistance. And this is an ongoing collaboration, and we're collaborating with essentially Daniel and also some other labs as well to try to try to use our antibody as a way of finding novel targets. So that's GLUT4, and I just want to end with some other challenges that have to do with transporters. So as I mentioned at the beginning, the SLC family is a very diverse family of transporters, and they have a lot of challenges. There's a lot of structural and functional diversity. They can have very small extracellular loops. The structures can be state specific, so that can actually be a challenge depending on what state you're looking for. But there's another challenge here as well, which is accessory subunits. And that's an unusual challenge. And let me show that here. So some SLC families have accessory subunits. And what do I mean by that? So I'm going to talk here, this is a recent structure of a transporter SLC7A5, also known as LAT1. SLC7A5 is a neutral amino acid transporter. It transports amino acids like leucine and tryptophan into the cell. And LAT1 is associated with another subunit, sometimes known as SLC3A2, also known as CD98HC, CD98 heavy chain. So it's a CD marker. And what you can see is LAT1, SLC7A5 in blue here, is what you would expect of as a transporter, and this is the part of the subunit that does all the transporter activity. On the other hand, there's this large extracellular domain, CD98HC, which almost completely covers the transporter. And what's interesting, when they did this structure, it was in the presence of an antibody against CD98. And you can see that it's easy for an antibody to bind the subunit, but it's really hard to imagine how an antibody can access the transporter part of this SLC7A5, because it's heavily occluded by this CD98 heavy chain, the SLC3A2 subunit. So the accessory subunits can block the relevant epitopes. And it's not just this transporter. There's a number of transporters that have these blocking, occluding subunits. But we can get around that. And how do we get around that? And we think it actually has to do with a lot to do with these chicken antibodies, with these long CDRs. And here's what we were able to do. most of the antibodies that we're able to get against this transporter, and it's not LAT1, it's a different transporter. I can't tell you which transporter it is. But what we're able to do is most of the antibodies, they bind the transporter alone just fine, and you can express this transporter in the absence of the accessory unit. But when you add the accessory unit, you completely lose binding of the antibody. So it's an antibody against the transporter, but in the presence of the subunit, you don't see binding. What we do, on the other hand, is we found some rare antibodies that were able to bind the transporter alone, and in the presence of the accessory unit, really we're almost able to bind the transporter in an identical fashion. Now, there are some other explanations for this as well, but I will say that we have additional data, which unfortunately I cannot share with you, that really shows that what this antibody is doing is accessing the transporter in a way that the accessory subunit is unable to occlude it. So it's binding a very, very restricted epitope. And we feel that our antibody discovery platform is really tailored to allow us to access these occluded epitopes. And we really do think it has a lot to do with chickens. We've routinely isolate functional antibodies using chicken immunization, and here are some examples. Here where we've gotten functional antibodies, antagonists, and antibodies and inhibitors against ion channels, GPCRs, and transporters. And what's unique about them is most of these have very, very unusually long HCDR3s, which once again is a hallmark of chicken antibodies. And we think that these long CDRs enable us to access novel epitopes, which often have functional activity. And we really think that, combined with our robust immune methodologies using lipoparticles and DNA, coupled with our expertise in panning using lipoparticles, enables us to pull out these rare antibodies. So to finish up, as I said, integral molecular, we do a lot of antibody discovery for partners, but we also have our own pipeline in a variety of different therapeutic areas, including metabolic disease, oncology, immune modulation, pain, and we're happy to talk to anybody about any of these programs if you have more questions. As I said, we work with many companies, over 300 companies, some of which I can talk about. It's public, and you can see them listed here. We're also been lucky that we've been able to publish both papers and patents on a lot of our technology, and so we're very much a science-driven company, and we love talking about science and papers and at conferences like this. So I want to thank you for your attention, and I'm more than happy to answer any of your questions in the chat. And thank you, and enjoy the rest of the conference.